Embryo sac development and double fertilization in plants. CSIR Net JRF Life Sciences Lecture. In angiosperms flowering plants, the processes of embryo sac development and double fertilization are pivotal to sexual reproduction and seed formation. The embryo sac is a female gametophyte that develops within the ovule of the ovary. Double fertilization is a unique event to angiosperms in which two sperm cells from the pollen grain fertilize two different cells within the embryo sac. One sperm fertilizes the egg to form the zygote, and the other fuses with the central cell to form the triploid endosperm. Both these processes are exquisitely coordinated and highly specialized. The embryo sac develops from a single megaspore mother cell, MMC, located within the nucellus of the ovule. This diploid cell undergoes meiosis to produce 4 haploid megaspores, of which usually only one becomes functional. This surviving megaspore undergoes 3 successive meiotic divisions without cytokinesis, resulting in 8 nuclei within a common cytoplasm. These nuclei then undergo cellular organization to form a 7-celled, 8-nucleate structure called the embryo sac. The 7 cells are organized into 3 major groups: the egg apparatus at the micropolar end, comprising 1 egg cell and 2 synergids, the central cell containing 2 polar nuclei, and 3 antipodal cells located at the chalazole end. This developmental pattern is referred to as the polygonum type, the most common type of embryo sac development in angiosperms. The central cell, being binucleate, plays a crucial role in double fertilization, while the synergids contain filiform apparatus, specialized structures that guide the incoming pollen tube and facilitate sperm delivery. The antipodal cells typically degenerate post-fertilization but may have nutritive or signaling roles during gametophyte development. The development of the embryo sac is precisely controlled by gene networks that regulate megasporogenesis, megagametogenesis, and cell phase specification. Mutations in genes such as BEL1, ANT, and NEA can lead to aberrant gametophyte formation and infertility. Fertilization in flowering plants is double and unique. It begins when a pollen grain lands on a compatible stigma, germinates, and grows a pollen tube through the style towards the ovule. The vegetative tube cell drives tube growth, while the generative cell divides mitotically to produce 2 non-motile sperm cells. The pollen tube is guided towards the embryo sac by chemoattractants, such as lure peptides, secreted by synergid cells. Upon reaching the micropyle, the pollen tube enters one of the synergids and bursts, releasing the 2 sperm cells. One sperm cell fuses with the egg cell to form the diploid zygote, 2N, initiating embryogenesis. The other sperm fuses with the 2 polar nuclei in the central cell to form the triploid primary endosperm nucleus, 3N, which develops into the endosperm, a nutritive tissue for the developing embryo. This process is termed double fertilization and ensures synchronized development of embryo and endosperm, which is crucial for seed viability. Following fertilization, synergids and antipodal cells degenerate, and the zygote undergoes a predictable pattern of cell divisions to form the embryo proper. The endosperm can develop in different ways: nuclear, most common, cellular, or halobial. Nuclear endosperm development begins with 3 nuclear divisions without cell wall formation, while cellular type forms walls after each division, and halobial is intermediate. This triploid endosperm serves as an immediate source of nutrition to the growing embryo in cereals and other seed plants. Double fertilization is an evolutionary innovation that is exclusive to angiosperms and absent in gymnosperms. It allows for efficient allocation of maternal resources by initiating endosperm development only upon successful fertilization. The molecular players involved include FusCA3, LEC1, and NEA, which regulate embryo and endosperm development. Disruption of double fertilization can result in seed abortion, often due to failure in pollen tube guidance, sperm cell release, or fusion processes. Furthermore, genes like GCS1, CAP2 are essential for gamete fusion, while pheronia and lorelei, expressed in synergids, are necessary for pollen tube reception and sperm cell release. Recent discoveries highlight the role of epigenetic mechanisms, such as DNA methylation and histone modifications, in regulating embryo sac developmentalization. Imprinting in endosperm genes like MEA ensures differential expression of maternal and paternal alleles, critical for proper seed development. Embryo sac development and double fertilization not only represent the complexity of plant sexual reproduction but also offer a framework for biotechnological applications. For instance, manipulation of these processes can lead to apomixis, a phenomenon where seeds are formed without fertilization, which has great potential for fixing hybrid vigor. Moreover, understanding these mechanisms is crucial for improving crop yields, controlling seed development, and enhancing reproductive success under stress conditions. In summary, embryo sac development in angiosperms follows a tightly regulated series of meiotic divisions and cellular differentiations leading to a 7-celled, 8-nucleate structure, while double fertilization involves 2 distinct fusion events within the sac that result in embryo and endosperm formation. This highly evolved process is unique to flowering plants, ensures efficient seed formation, and is governed by intricate genetic, molecular, and epigenetic regulatory networks. Previous year questions: CSIR Net JRF Life Sciences. CSIR Net JRF December 2015. Which of the following correctly depicts the typical angiosperm embryo sac? Answer: 7 cells, 8 nuclei. Explanation: the mature embryo sac in angiosperms consists of 7 cells, egg, 2 synergids, 3 antipodals, and 1 central cell and 8 nuclei. Central cell contains 2 nuclei. CSIR Net JRF June 2016. What is the ploidy level of endosperm in flowering plants after double fertilization? Answer: triploid 3N. Explanation: 1 sperm fuses with 2 polar nuclei forming the triploid endosperm nucleus. CSIR Net JRF December 2017. The pollen tube releases 2 sperm cells into which part of the embryo sac? Answer: one of the synergid cells. Explanation. The pollen tube enters the synergid, which degenerates and facilitates sperm release for fertilization. Let me know the next topic, and I'll continue preparing the next CSIR Net JRF level lecture for you in the same format. You said, zygote formation, ChatGPT said. The pollen tube releases two sperm cells into which part of the embryo sac? Answer, one of the synergid cells. Explanation. The pollen tube enters the synergid, which degenerates and facilitates sperm release for fertilization. Let me know the next topic, and I'll continue preparing the next CSIR Net JRF level lecture for you in the same format. You said, zygote formation, ChatGPT said. Here is your CSIR Net JRF Life Sciences High-Level Theory Lecture on zygote formation, written in maximum word limit, paragraph format, no tables, and including 3 CSIR Net previous year questions with answers in year mentioned. This lecture is meticulously crafted to serve as your only and complete study material, filled with exam-relevant points from base to advanced level, tailored to CSIR Net. Zygote formation, CSIR Net JRF Life Sciences Detailed Lecture. Zygote formation is a central event in sexual reproduction, marking the successful fusion of male and female gametes to restore diploidy and initiate a new individual. In all sexually reproducing eukaryotes, the zygote is the first cell of the next generation, formed as a result of syngamy, the process of gamete fusion. The formation of the zygote is not merely a fusion of genetic materials but involves highly orchestrated cellular, molecular, and structural changes that ensure the success of embryogenesis. Zygote formation is universally conserved across animals and plants but with important mechanistic differences, particularly in the context of cellular mechanisms and developmental cues. In animals, zygote formation begins when a haploid sperm cell fuses with a haploid egg cell, resulting in a diploid zygote 2N. The fusion process involves several well-defined steps: species-specific sperm egg recognition, membrane fusion, activation of the egg, and pronuclear fusion. Upon sperm binding, molecules such as Azuma 1 on the sperm surface interact with Juno on the oocyte membrane, initiating the fusion. Following this, the egg undergoes cortical granule exocytosis, releasing enzymes that modify the zona pellucida to prevent polyspermy, the entry of additional sperm. This is essential because multiple sperm nuclei would cause lethal polyploidy. Egg activation is triggered by a rapid rise in intracellular Ca² concentration, often initiated by PLC zeta from the sperm, which restarts the arrested oocyte at metaphase 2 of meiosis in mammals. Once the sperm nucleus enters the egg, the paternal DNA is decondensed, forming the male pronucleus, while the maternal genome completes its second meiotic division and forms the female nucleus. These two pronuclei migrate towards each other and their envelopes dissolve during syngamy, allowing the mixing of maternal and paternal chromosomes at the metaphase plate of the first zygotic meiotic division. The result is a genetically unique diploid zygote, containing a combination of alleles from both parents. The zygote is totipotent, capable of giving rise to all cell types of the organism, including extraembryonic tissues in many animals. In plants, zygote formation also follows the fusion of haploid gametes, but with unique aspects. In angiosperms, the egg cell is located in the embryo sac, and the male gametes sperm cells are delivered through the pollen tube. As part of double fertilization, one sperm cell fuses with the egg cell to form the diploid zygote, while the other sperm fuses with the two polar nuclei to form the triploid endosperm nucleus. The egg and sperm plasma membranes fuse, followed by fusion of the gamete nuclei karyogamy. The zygote remains quiescent for a brief period before entering asymmetric division to form the apical cell, which becomes the embryo, and the basal cell, which becomes the suspensor. Molecular components such as FusCA3, WOX2, WOX8, and YDA signaling pathways regulate this early zygotic development in plants. Zygote formation is not just a mechanical union of gametes but a reprogramming event. In angiosperms, the egg cell is located in the embryo sac, and the male gametes sperm cells are delivered through the pollen tube. As part of double fertilization, one sperm cell fuses with the egg cell to form the diploid zygote, while the other sperm fuses with the two polar nuclei to form the triploid endosperm nucleus. In angiosperms, the egg cell is located in the embryo sac, and the male gametes sperm cells are delivered through the pollen tube. As part of double fertilization, one sperm cell fuses with the egg cell to form the diploid zygote, while the other sperm fuses with the two polar nuclei to form the triploid endosperm nucleus. In angiosperms, the egg cell is located in the embryo sac, and the male gametes sperm cells are delivered through the pollen tube. As part of double fertilization, one sperm cell fuses with the egg cell to form the In angiosperms, the egg cell is located in the embryo sac, and the male gametes sperm cells. In angiosperms, the egg cell is located in the embryo sac, and the male gametes sperm cells are delivered through the pollen tube. As part of double fertilization, one sperm cell fuses with the egg cell to form the diploid zygote, while the other sperm fuses with the two polar nuclei to form the triploid endosperm nucleus. The egg and sperm plasma membranes fuse, followed by fusion of the gamete nuclei karyogamy. The zygote remains quiescent for a brief period before entering asymmetric division to form the apical cell, which becomes the embryo, and the basal cell, which becomes the suspensor. Molecular components such as FusCA3, WOX2, WOX8, and YDA signaling pathways regulate this early zygotic development in plants. Zygote formation is not just a mechanical union of gametes but a reprogramming event. In mammals, extensive epigenetic reprogramming occurs, including DNA demethylation, histone replacement, and chromatin remodeling to establish the In mammals, extensive epigenetic reprogramming occurs, including DNA demethylation, histone replacement, and chromatin remodeling to establish the totipotent state. The maternal cytoplasm contains critical factors like maternal mRNAs, RNAs, and proteins that drive early development until zygotic genome activation, ZGA, takes over, typically at the 2-cell stage in mice and 8-cell stage in humans. In plants, the zygotic genome is activated almost immediately, and zygote polarity is established through uneven distribution of transcription factors and hormones like auxin. Defects in zygote formation can lead to infertility, early embryonic arrest, or developmental abnormalities. For instance, mutations in HAP2, GCS1, a sperm-expressed fusion protein, can prevent gamete fusion in plants and protists. Similarly, in humans, mutations in PLC zeta, Azuma 1, or Juno can result in failed fertilization and zygote formation. Research in zygote formation has important implications in assisted reproductive technologies, such as in vitro fertilization, IVF, and zygote intrafallopian transfer, ZIFT. Understanding how the zygote forms and activates its genome has also enhanced our understanding of stem cell biology, reprogramming, and early lineage specification. In conclusion, zygote formation is a central, conserved, and intricately regulated process in the life cycle of all sexually reproducing organisms. It ensures the combination of genetic material from two parents, reestablishes diploidy, and initiates the program of development through complex cellular and molecular events. In plants and animals alike, this marks the beginning of a new organism, equipped with totipotency and developmental potential, making it a critical topic in developmental biology and reproductive science. Previous year questions: CSIR Net JRF Life Sciences. CSIR Net JRF December 2013. Which of the following molecules is essential for sperm egg membrane fusion in mammals? Answer: Azuma 1. Explanation: Azuma 1 on sperm binds to Juno on the egg membrane and is essential for fusion. CSIR Net JRF June 2018. During fertilization in flowering plants, which cell fuses with the egg to form the zygote? Answer. One of the sperm cells. Explanation. Of the two sperm cells released, one fuses with the egg to form the zygote. CSIR net JRF December 2019. What is the ploidy of the zygote formed after fertilization in angiosperms? Answer. Diploid 2N. Explanation. Zygote is formed by fusion of one haploid sperm with a haploid egg. Let me know your next topic and I'll prepare the next perfect lecture immediately for you. You said, cleavage, ChatGPT said. Here is your CSIR net JRF Life Sciences top tier lecture on cleavage, written in maximum word limit, paragraph format, with no tables, and incorporating 3 previous year CSIR net questions with answers and years mentioned. This is a comprehensive, fully loaded, and exam-oriented theory note designed to be your only and complete study material for cracking the CSIR net exam. Every line is packed with crucial information, no free space is wasted. Cleavage, CSIR net JRF Life Sciences high yield lecture. Cleavage is the process of a series of rapid meiotic divisions of the zygote that transforms the single-celled fertilized egg into a multicellular embryo without overall growth in size. It marks the first phase of embryonic development following fertilization and results in the formation of a solid ball of smaller cells known as blastomeres. The end product of cleavage is typically a blastula, a hollow or solid ball of cells, depending on the species. Cleavage is fundamentally controlled by maternal factors deposited in the oocyte, and it proceeds before the activation of the zygotic genome in most animals. Cleavage divisions are unique because they do not involve significant cell growth. The G1 and G2 phases of the cell cycle are either extremely short or absent, allowing the embryo to rapidly increase the number of cells. These divisions are highly synchronized in early stages and driven by maternal mRNAs and proteins, including cyclins and cyclin-dependent kinesis CDKs. In animals such as amphibians and mammals, MPF maturation promoting factor and CDC25 phosphatase play key roles in regulating the timing and progression of these divisions. The pattern of cleavage depends on several factors, including the amount and distribution of yolk, vitaline content, the orientation of meiotic spindles, and the location of centrosomes. In isolecithal eggs, sparse yolk, evenly distributed as seen in echinoderms and mammals, cleavage is holoblastic complete and usually radial or rotational. In mesolecithal eggs, moderate yolk concentrated at the vegetal pole, such as in amphibians, cleavage is still holoblastic but displaced due to yolk-hindering cytokinesis in the vegetal hemisphere. In contrast, telolecithal eggs, dense yolk at vegetal pole, like in birds and reptiles, exhibit meroblastic incomplete cleavage, typically discoidal, restricted to a small cytoplasmic region. Centrolecithal eggs, yolk in center, as in insects, show superficial cleavage, where nuclear divisions occur without cytokinesis, forming a syncytium before cellularization. Based on symmetry and orientation, cleavage can be categorized as radial, spiral, bilateral, rotational, and discoidal. For instance, radial cleavage is observed in echinoderms and is characterized by perpendicular or parallel cleavage planes relative to the polar axis. Spiral cleavage, seen in annelids and mollusks, involves oblique meiotic spindles resulting in twisted blastomeres. Rotational cleavage is characteristic of mammals, where the first division is meridional and the second is unequal, one meridional and one equatorial, leading to asynchronous cleavage. Cleavage also sets the stage for the first cell fate decisions, especially in mammals. Around the 8-cell stage, mammalian embryos undergo compaction, where blastomeres become tightly adherent due to E-cadherin-mediated cell adhesion, leading to the formation of inner and outer cell populations. These form the inner cell mass ICM and trophoblast, respectively, which later contribute to the embryo proper and placenta. October 4, NANOG, and CDX2 are key transcription factors involved in the differentiation of these lineages during. In amphibians, cleavage divisions help localize cytoplasmic determinants like vectin BG1 in the vegetal hemisphere and WNT11 in the dorsal region, essential for axis formation. In mammals, cleavage is slower and asynchronous compared to other vertebrates, and the embryo remains encased within the zone of Bleuler. At the Marola stage, 16 to 32 cells, the embryo prepares for blastocyst formation, where fluid-filled cavities coalesce to form the blastocele. The timing of zygotic genome activation (ZGA) is also species-specific. In mice, it begins at the 2-cell stage, whereas in humans, it starts at the 8-cell stage. The regulation of cleavage also involves epigenetic reprogramming, including DNA demethylation and histone modifications, which reset the developmental potential of the embryo. In invertebrates, cleavage patterns are often deterministic, with clear cell lineage maps, mosaic development, as in C. elegans. In contrast, vertebrates like mammals and amphibians show regulated development, where cell fates are more flexible during early cleavage stages. This flexibility forms the basis of embryonic stem cell totipotency and pluripotency, making cleavage an essential stage for understanding early developmental plasticity. In conclusion, cleavage is a critical stage of embryonic development characterized by rapid, synchronous cell divisions that partition the zygote into smaller, genetically identical cells while preserving the overall size of the embryo. The patterns and molecular regulation of cleavage determine the early architectural layout of the embryo and are essential for proper cell fate specification, axis formation, and blastula formation. Understanding cleavage not only sheds light on basic developmental biology but also forms the foundation for modern stem cell research, cloning, and assisted reproductive technologies. Previous year questions. CSIR net JRF Life Sciences. CSIR net JRF June 2015. Which of the following statements about cleavage in mammals is correct? Answer. Cleavage is asynchronous and results in compaction by the 8-cell stage. Explanation. In mammals, cleavage is slow and asynchronous, and compaction occurs at the 8-cell stage via E-cadherin. CSIR net JRF December 2014. In which type of eggs does discoidal meroblastic cleavage occur? Answer. Telolecithal eggs. Explanation. Birds and reptiles have telolecithal eggs where cleavage is restricted to small disc on the yolk. CSIR net JRF June 2019. What type of cleavage pattern is exhibited by echinoderms? Answer. Holoblastic radial cleavage. Explanation. Echinoderm embryos show complete and radial cleavage, typical of isolecithal eggs. Let me know your next topic, e.g., blastula formation or gastrulation, and I'll prepare the next full scope lecture for you right away. You said, blastula formation ChatGPT said. Here is your CSIR net JRF Life Sciences exam level lecture on blastula formation, written in maximum word limit, highly detailed, paragraph only format, with no tables, and including 3 previous year CSIR net questions with answers and years mentioned. This note is constructed with the precision and depth required to be your sole, complete coaching material, focused on previously asked areas and important concepts. No free space is wasted. Blastula formation, CSIR net JRF Life Sciences maximum detail lecture. Blastula formation is the developmental stage that immediately follows cleavage in animal embryogenesis. It marks the transition from a solid or loosely arranged mass of blastomeres into a structured, often hollow, sphere of cells surrounding a fluid-filled cavity known as the blastocele. The blastula is a pivotal embryonic stage, preparing the embryo for gastrulation and establishing the first signs of regional specification and germ layer separation. The form and structure of the blastula vary among species and are highly influenced by the amount and distribution of yolk in the egg. In organisms with isolecithal eggs like sea urchins and mammals, cleavage is holoblastic and leads to a coeloblastula, which is a hollow ball of cells surrounding a centrally located blastocele. In these organisms, all blastomeres contribute equally to the formation of the blastula, and the cells remain relatively small due to complete cleavage. In contrast, mesolecithal eggs, such as those of amphibians, exhibit unequal holoblastic cleavage, resulting in a blastula with a displaced blastocele. The larger vegetal blastomeres divide more slowly and contribute less to the blastocele lining, leading to an eccentric cavity closer to the animal pole. In telolecithal eggs, such as those of birds and reptiles, cleavage is meroblastic and discoidal, leading to the formation of a discoblastula. In organisms with isoleucical eggs like sea urchins and mammals, cleavage is holoblastic and in organisms with isoleucical eggs like sea urchins and mammals, cleavage is holoblastic and leads to a coaloblastula, which is a In organisms with isoleucical eggs like sea urchins and mammals, cleavage is holoblastic and leads to a coaloblastula, which is a hollow ball of cells surrounding a centrally located blastocele. In these organisms, all blastomeres contribute equally to the formation of the blastula, and the cells remain relatively small due to complete cleavage. In contrast, mesoleucical eggs, such as those of amphibians, exhibit unequal holoblastic cleavage, resulting in a blastula with a displaced blastocele. The larger vegetable blastomeres divide more slowly and contribute less to the blastocele lining, leading to an eccentric cavity closer to the animal pole. In teloleucical eggs, such as those of birds and reptiles, cleavage is narrowblastic and discoidal, leading to the formation of a discoblastula. Here, only the small disc of cells sitting atop the yolk mass contributes to the blastula, and the blastocele lies between the cell layer epiblast and the underlying yolk hypoblast. The mammalian blastula is a special structure known as a blastocyst, formed following the Marola stage. During blastocyst formation, compaction and fluid transport via Nau+, K+ ATPase pumps generate a blastocele. The outer layer of cells, known as the trophoblast, contributes to the placenta, while the inner cell mass ICM gives rise to the embryo proper. Blastula formation is not merely a morphological transformation, it involves crucial molecular and cellular arrangements. During this phase, the mid-blastula transition MBT occurs in many species, especially invertebrates like amphibians. At MBT, the zygotic genome is activated, and transcription of embryonic genes begins, replacing maternal mRNAs. Additionally, cell cycles lengthen with the addition of G1 and G2 phases, cell motility increases, and asynchronous divisions begin. This phase is marked by the initiation of cell differentiation and fate specification. Key regulatory factors during MBT include Zelda in Drosophila and October 4, SOX2, and Nanog in mammals. The blastula also displays the first signs of polarity and regional specification, crucial for later gastrulation. In amphibians, the animal-vegetal axis becomes well-defined, with prospective ectoderm forming in the animal region and endodermal precursors located vegetally. In amphibians and echinoderms, cells in the vegetal region also contain determinants such as beta-catenin, which activate the formation of the organizer region or dorsal lip of the blastopore during gastrulation. The differential distribution of these determinants is essential for germ layer formation and axis patterning. In mammals, the formation of the epiblast and hypoblast primitive endoderm from the ICM during blastocyst maturation is a critical lineage segregation step. This is guided by signaling pathways like FGF-ERK, where FGF4 from epiblast cells promotes hypoblast fate. Epiblast cells exhibit apico-basal polarity, which is crucial for the subsequent formation of the amniotic cavity and primitive streak. This functional specification within the blastula is vital for proper gastrulation movements and formation of the three germ layers. Moreover, cell adhesion molecules play a vital role during blastula formation. Proteins like E-cadherin mediate compaction and adhesion between blastomeres, while tight junctions and gap junctions ensure communication and polarity. The establishment of these junctions is important for cavity formation and for maintaining the integrity of the epithelium during morphogenic movements. The blastula stage also allows for experimental manipulation, such as fate mapping and embryonic transplantation, due to the regulated nature of the embryo at this stage. For example, blastula stage transplantation experiments in xenopus have been instrumental in identifying the role of the semen organizer, while in mammals, embryonic stem cells isolated from the ICM at this stage retain pluripotency and can be used for cloning and regenerative medicine. In conclusion, the blastula represents a highly organized, molecularly dynamic stage of early embryogenesis. It prepares the embryo for the complex movements and cell fate decisions of gastrulation, establishes key cell lineages, activates the embryonic genome, and reflects the earliest regional identities of the future body plan. Understanding the variations in blastula structure among different taxa and the molecular mechanisms underlying its formation is essential for mastering developmental biology and successfully answering high-level questions in CSIR-NET JRF Life Sciences. Previous year questions, CSIR-NET JRF Life Sciences. CSIR-NET JRF December 2013. Which of the following statements is true regarding the blastocyst of mammals? Answer: the trophoblast forms the outer epithelium and contributes to placental formation. Explanation: the outer layer of the mammalian blastocyst is the trophoblast, which later participates in forming the placenta, while the ICM forms the embryo. CSIR-NET JRF June 2016. At which stage of development does zygotic genome activation typically occur in vertebrates? Answer: mid-blastula transition. Explanation: the mid-blastula transition is characterized by zygotic genome activation, lengthened cell cycles, and onset of transcription. CSIR-NET JRF June 2018. What is the role of Nau+, K+? Explanation: the mid-blastula transition is characterized by zygotic genome activation, lengthened cell cycles, and onset of transcription. CSIR-NET JRF June 2018. What is the role of Nau+, K+ ATPase during blastocyst formation in mammals? Answer: it helps in forming the blastocele by creating osmotic gradients for fluid accumulation. Explanation: the pump actively transports sodium ions into the cavity, drawing water osmotically to expand the blastocele. Let me know when you're ready for the next topic, e.g., gastrulation, and I will continue with the same perfect format. You said, embryonic fields, ChatGPT said. Here is your CSIR-NET JRF Life Sciences ultra detailed, paragraph-based lecture on embryonic fields, written with maximum word limit, no tables, and covering base to advanced concepts. This lecture includes 3 previous year CSIR-NET questions with answers in years mentioned, and is tailored to be your soul coaching material, filled with highly exam-relevant theory based on previous question trends, with zero space wasted. Embryonic fields, CSIR-NET JRF Life Sciences maximum detail lecture. Embryonic fields, also called morphogenic fields, are defined as discrete regions of the embryo within which specific cell populations are predestined to give rise to particular tissues or organs. These fields represent functional and spatial units during early development where cells communicate, respond to positional information, and execute coordinated developmental programs to form complex structures. The concept of embryonic fields arises from classical embryology, particularly the work of Hans Semmen and others, who demonstrated that certain regions of the embryo possess an intrinsic developmental potential that can be reorganized or reassigned when transplanted, yet still form complete structures, revealing their regulative capacities. Each embryonic field contains determined cells, inductive signals, and morphogen gradients, which work in concert to direct developmental fate. Importantly, these fields are not fixed early on but exhibit plasticity. Cells within a field can still change fate if exposed to a different signaling environment or if isolated. As development progresses, this plasticity decreases, and cells within a field become fate restricted. The presence of organizers, such as the Semmen organizer in amphibians, further patterns embryonic fields by secreting signaling molecules like noggin, chortin, and follistatin, which modulate BMP signaling and define neural and mesodermal fields. Embryonic fields are evident in the early mesoderm, where prospective heart, notochord, somites, and kidney fields become evident. The cardiogenic field is a classic example. A bilateral region of splanchnic mesoderm adjacent to the primitive streak gives rise to the heart. Transplantation studies have shown that even if parts of the field are removed, the remaining cells can compensate and still form a functional heart, demonstrating the self-regulating nature of embryonic fields. However, if the entire field is ablated, no heart develops, indicating the field's autonomy. Similarly, the limb field demonstrates regional identity and patterning potential. The limb field is established in the lateral plate mesoderm in association with overlying ectoderm, under the influence of Hox gene expression gradients. Transplanting limb field cells to ectopic sites can induce limb-like outgrowths, proving their positional identity. This field also demonstrates the role of positional information in gradients of FGF, SHISH, and retinoic acid, which help establish the proximal distal. Embryonic fields, also called morphogenic fields, are defined as discrete regions of the embryo within which specific cell populations are predestined to give rise to particular tissues or organs. Fields represent functional and spatial units during early development where cells communicate, respond to positional information, and execute coordinated developmental programs to form complex structures. The concept of embryonic fields arises from classical embryology, particularly the work of Hans Femen and others, who demonstrated that certain regions of the embryo possess an intrinsic developmental potential that can be reorganized or reassigned when transplanted, yet still form complete structures, revealing their regulative capacities. Each embryonic field contains determined cells, inductive signals, and morphogen gradients, which work in concert to direct developmental fate. These fields are not fixed early on but exhibit plasticity. Cells within a field can still change fate if exposed to a different signaling environment or if isolated. As development progresses, this plasticity decreases, and cells within a field become fate restricted. The presence of organizers, such as the Femen organizer in amphibians, further patterns embryonic fields by secreting signaling molecules like noggin, chordin, and follistatin, which modulate BMP signaling and define neural and mesodermal fields. Embryonic fields are evident in the early mesoderm, where prospective heart, notochord, somites, and kidney fields become evident. The cardiogenic field is a classic example. A bilateral region of splanchnic mesoderm adjacent to the primitive streak gives rise to the heart. Transplantation studies have shown that even if parts of the field are removed, the remaining cells can compensate and still form a functional heart, demonstrating the self-regulating nature of embryonic fields. However, if the entire field is ablated, no heart develops, indicating the field's autonomy. Similarly, the limb field demonstrates regional identity and patterning potential. The limb field is established in the lateral plate mesoderm in association with overlying ectoderm, under the influence of Hox gene expression gradients. Transplanting limb field cells to ectopic sites can induce limb-like outgrowths, proving their positional identity. This field also demonstrates the role of positional information in gradients of FGF, SHISH, and retinoic acid, which help establish the proximal distal, anterior posterior, and dorsal ventral axes within the developing limb. At the molecular level, embryonic fields are governed by networks of transcription factors, signaling molecules, and feedback loops that maintain field integrity. For example, in neural development, the anterior neural field is regulated by WNT antagonists like DKK1 and Cerberus, which promote forebrain identity, while higher WNT activity posteriorizes the neural field. This compartmentalization within the neural field leads to the establishment of the This field also demonstrates the role of positional information in gradients of FGF, SHISH, and retinoic acid, which help establish the proximal distal, anterior posterior, and dorsal ventral axes within the developing limb. This field also demonstrates the role of positional information in gradients of FGF, SHISH, and retinoic acid, which help establish the proximal distal, anterior posterior, and dorsal ventral axes within the developing limb. At the molecular level, embryonic fields are governed by networks of transcription factors, signaling molecules, and feedback loops that maintain field integrity. For example, in neural development, the anterior neural field is regulated by WNT antagonists like DKK1 and Cerberus, which promote forebrain identity, while higher WNT activity posteriorizes the neural field. This compartmentalization within the neural field leads to the establishment of the forebrain, midbrain, hindbrain, and spinal cord regions, each with its own subfield and patterning mechanisms. The interaction between adjacent fields is another essential feature. Boundaries between fields are maintained through differential gene expression and cell adhesion molecules, such as E-cadherin and N-cadherin, and through lineage restriction boundaries like the midbrain-hindbrain boundary isthmic organizer. These boundaries are critical to prevent mixing of fate-specified populations and to maintain coherent organogenesis. In vertebrates, cell lineage tracing and fate mapping studies using dyes, genetic reporters, and clonal analysis have enabled precise identification of fields. Such studies have demonstrated that within a given field, cells respond collectively to gradients of morphogens like BMPs, FGFs, WNTs, and hedgehog proteins, which diffuse from signaling centers to regulate gene expression in a concentration-dependent manner. This principle underlies the concept of morphogenic gradients, where a single morphogen can induce multiple fates across a field, depending on its local concentration. In summary, embryonic fields represent spatially and functionally organized regions of an embryo that harbor the coordinated capacity to form discrete tissues or organs. They are defined by regulatory gene expression, inductive signals, and morphogen gradients, and they serve as the structural and functional basis for organogenesis. Their plasticity, regulatory autonomy, and molecular compartmentalization are central to understanding developmental processes and are of immense importance in experimental embryology, regenerative medicine, and evolutionary developmental biology. Mastery of this topic provides a powerful conceptual framework for approaching questions on axis formation, fate mapping, morphogenic movement, and organ development in the CSIR-NET JRF Life Sciences exam. Previous year questions: CSIR-NET JRF Life Sciences. CSIR-NET JRF June 2017. Which of the following best describes the concept of embryonic fields? Answer: regions of an embryo where cells are committed to form specific tissues and can regulate development when part of the field is damaged. Explanation: embryonic fields are areas with self-regulated properties and specific developmental fate. CSIR-NET JRF December 2015. Transplantation of the limb bud field into a different location in the embryo results in the formation of a limb-like structure. Does this indicate? Answer: cells in the field are specified and can express their identity autonomously. Explanation: limb fields contain cells with positional and patterning information, capable of generating limbs even ectopically. CSIR-NET JRF June 2020. What is the role of morphogen gradients within embryonic fields? Answer: they provide positional information to pattern the tissue based on concentration thresholds. Explanation: morphogens like SHISH and BMPs act in a concentration-dependent manner to induce diverse fates within a field. Let me know when you're ready for the next topic, such as gastrulation, and I'll continue with the same high standard. You said, gastrulation. ChatGPT said. Here is your CSIR-NET JRF Life Sciences Ultra Detailed Lecture on Gastrulation, crafted exactly as you requested, maximum word limit, paragraph form, no tables, no wasted space, and tailored from base to advanced level, with 3 previous year CSIR-NET questions, answers, and years mentioned. This is designed to be your only coaching material and is fully aligned with CSIR-NET JRF standards based on repeated question areas. Gastrulation, CSIR-NET JRF Life Sciences High Yield Theory. Gastrulation is the foundational morphogenic process in embryonic development during which a single-layered blastula is reorganized into a multi-layered structure known as the gastrula. This process leads to the formation of the 3 primary germ layers, ectoderm, mesoderm, and endoderm, which will later give rise to all tissues and organs of the organism. Gastrulation represents the beginning of morphogenesis and establishes the body plan, including the anterior posterior, dorsal ventral, and left-right axes of the embryo. Gastrulation is marked by coordinated cell movements, including invagination, involution, epiboly, ingression, and delamination. These movements reshape the embryo and allow the internalization of cells that will give rise to the embryo. Although the ultimate goal of gastrulation is conserved across species, the mechanics and timing vary among vertebrates. For instance, in amphibians such as xenopus, gastrulation begins with the formation of the dorsal lip of the blastophore, known as the Femen organizer, which initiates a series of inductive events. The organizer secretes inhibitors of BMP signaling, e.g., noggin, chordin, follistatin, to promote neural induction and dorsalization of the mesoderm. In birds and mammals, gastrulation involves the formation of the primitive streak, a structure formed on the epiblast through which epiblast cells ingress to form mesoderm and endoderm. The anterior end of the primitive streak forms the Hensen's node, the equivalent of the Femen organizer, which emits signals that pattern the notochord and define the embryonic midline. The apoplast gives rise to all three germ layers, while the hypoblast is displaced and replaced by definitive endoderm. In mammals, the extraembryonic tissues play additional regulatory roles, releasing signals such as WNT3 and nodal to initiate streak formation. In sea urchins, gastrulation begins with primary mesenchyme cells ingressing into the blastocele, followed by invagination at the vegetal pole to form the archenteron, which eventually connects to the exterior via the blastopore. In Drosophila, a model invertebrate, gastrulation begins with the formation of a ventral furrow and invagination of mesodermal precursors, guided by genes like twist and snail. Despite morphological differences, all organisms show conserved molecular pathways regulating gastrulation, particularly the roles of TGF-beta nodal, WNT, FGF, and BMP signaling cascades. A critical feature of gastrulation is epithelial to mesenchymal transition, EMT, a process where tightly connected epithelial cells acquire migratory mesenchymal characteristics to move into the embryo's interior. EMT is regulated by transcription factors such as snail, slug, and twist, which repress adhesion molecules like E-cadherin, allowing cells to detach and migrate. This transition is vital for mesoderm and endoderm formation and sets the stage for further tissue specification. The germ layers established during gastrulation have specific fates. The ectoderm forms the nervous system and skin, the mesoderm forms muscle, bone, blood, and kidneys, while the endoderm gives rise to the gut, liver, pancreas, and respiratory tract. Fate mapping studies using fluorescent dyes and molecular tracers have confirmed the spatial allocation of these layers during and after gastrulation, providing critical insight into developmental patterning. Gastrulation is tightly regulated by morphogen gradients, especially of nodal for mesoderm induction BMP4, for ventralization WNT3A, for streak initiation, and FGFs for mesoderm migration. The timing, location, and intensity of these signals are tightly controlled, often through feedback loops, inhibitors e.g., lefty for nodal and cross-regulatory networks. Errors in gastrulation can lead to severe developmental anomalies such as gastrulation arrest, conjoined twins, or posterior truncation. In conclusion, gastrulation is a highly conserved yet species-specific process crucial for establishing the germ layers and embryonic axes. Its regulation involves a complex interplay of cell movements, signaling pathways, and transcriptional programs that coordinate spatial and temporal developmental events. Understanding gastrulation provides a foundation for studying organogenesis, pattern formation, and congenital anomalies, and remains a frequent and fundamental area tested in the CSIR-NET JRF Life Sciences examination. Previous year questions: CSIR-NET Life Sciences. CSIR-NET JRF June 2015. Which following events is associated with epithelial to mesenchymal transition during vertebrate gastrulation? Answer: loss of E-cadherin expression and acquisition of migratory phenotype. Explanation: EMT allows cells to ingress and migrate to form mesoderm and endoderm by repressing adhesion and enhancing motility. CSIR-NET JRF December 2018. In mammalian embryos, which structure functions as the organizer equivalent to Spemen's dorsal lip? Answer: Hen's node. Explanation: Hensen's node in mammals performs functions similar to the Spemen organizer, including axis formation and notochord induction. CSIR-NET JRF June 2021. Which link molecule is essential for primitive streak formation in mammalian gastrulation? Answer: No. Explanation: Nodal, a TGF-beta family member, is crucial for streak initiation and mesoderm induction. Let me know when you're ready for the next topic, neurulation, and I'll continue the same standard of comprehensive lecture writing. You said. Formation of germ layers in animals, ChatGPT said. Here is your CSIR-NET JRF Life Sciences Ultra Detailed Lecture on Formation of Germ Layers in Animals, written in maximum word limit, paragraph form, no tables, and tailored from base to advanced level, designed to be your only coaching material. It includes 3 previous year CSIR-NET questions with answers and years mentioned to ensure alignment with the exam's focus areas. This is crafted to cover all essential topics comprehensively. Formation of germ layers in animals, CSIR-NET JRF Life Sciences High Yield Theory. The formation of germ layers is a crucial developmental process that takes place during embryogenesis. It is the event in which the initial, indifferentiated cells of the blastula reorganize into 3 primary germ layers, ectoderm, mesoderm, and endoderm. These germ layers give rise to all tissues and organs in the animal body. The process of germ layer formation begins after gastrulation and sets the stage for the complex patterning of the organism. Understanding the mechanisms behind the formation and specification of these germ layers is critical for comprehending developmental biology, tissue differentiation, and organogenesis. The ectoderm is the outer layer of the embryo. It is responsible for forming the nervous system, including the brain and spinal cord, epidermis, the outer layer of the skin, and various structures derived from neural crest cells, such as melanocytes, facial bones, and parts of the heart. The ectoderm also contributes to the formation of sensory organs like the eyes and ears. The neural ectoderm, specifically, forms from the neural plate, which is induced by signals from the underlying notochord and precordial mesoderm. In vertebrates, this process is regulated by morphogens like sonic hedgehog, CHISH, WNT, and BMP signaling pathways. The mesoderm is the middle germ layer and gives rise to a variety of tissues, including the muscle, skeleton, cardiovascular system, and kidneys. It also gives rise to the gonads, testes, and ovaries, and the blood. During the process of gastrulation, cells from the epiblast ingress through the primitive streak and form the mesodermal layer, which undergoes further specification into paraxial mesoderm, giving rise to somites, intermediate mesoderm, giving rise to kidneys and gonads, and lateral plate mesoderm, giving rise to the heart and blood vessels. The formation of the mesoderm is guided by the expression of key signaling molecules such as nodal, WNT, and FGF, which help pattern the mesoderm along the anterior-posterior axis. The endoderm is the innermost germ layer and is crucial for the formation of the digestive tract, liver, lungs, and other internal organs. Cells of the epiblast ingress through the primitive streak to form the definitive endoderm, which gives rise to the lining of the gastrointestinal tract and respiratory system. The endodermal progenitor cells also contribute to the formation of the pancreas, thyroid, and parathyroid glands. The specification of the endoderm is regulated by signaling molecules such as nodal and BMP, with inhibition of WNT signaling being critical for definitive endoderm formation. During the formation of these 3 germ layers, complex molecular signaling pathways orchestrate their specification, patterning, and differentiation. One of the key signaling pathways in germ layer specification is the TGF-beta nodal signaling pathway, which plays a central role in mesoderm and endoderm induction. WNT signaling is also critical, with its activation promoting mesodermal fates, while inhibition of WNT signaling contributes to ectoderm formation. Additionally, BMP signaling is important for ectodermal patterning and mesodermal specification, but its activity must be tightly regulated during development to ensure proper germ layer formation. The molecular regulation of these pathways ensures that cells within the early embryo are appropriately positioned and specified according to their regional identity. For example, Hox genes play an essential role in establishing the anterior-posterior axis and patterning of the mesoderm and ectoderm. In combination with signaling molecules like FGF, BMP, and CHISH, Hox genes help to pattern the developing embryo by determining the identity of cells along the body axis. The process of germ layer formation in animals is not only crucial for the development of individual organs but also for the proper organization of tissues within the body. This organization ensures that the correct cell types are placed in the right locations, enabling the complex interaction and function of organs and systems. The concept of organizers, such as the Spemen organizer in amphibians and Hensen's node in mammals, highlights the importance of specific regions within the embryo that coordinate and regulate the development of surrounding tissues through the release of signaling factors. Furthermore, epithelial to mesenchymal transition (EMT) is a key phenomenon during the formation of the mesoderm and endoderm. Cells in the epiblast undergo EMT to become mesodermal or endodermal precursors, and this transition is regulated by the expression of specific transcription factors such as snail, twist, and slug. These transcription factors repress cell adhesion molecules like E-cadherin, allowing cells to migrate and invade the interior of the embryo. In conclusion, the formation of germ layers is a fundamental process that dictates the fates of cells and tissues during embryonic development. The ectoderm, mesoderm, and endoderm each give rise to distinct and critical structures, and their formation is regulated by a series of molecular signaling pathways and transcription factors. In conclusion, the formation of germ layers is a fundamental process that dictates the fates of cells and tissues during embryonic development. The ectoderm, mesoderm, and endoderm each give rise to distinct and critical structures, and their formation is regulated by a series of molecular signaling pathways and transcription factors. Formation of germ layers in animals. CSIR-NET-JRF Life Sciences High Yield Theory. The formation of germ layers is a crucial developmental process that takes place during embryogenesis. It is the event in which the initial, indifferentiated cells of the blastula reorganize into three primary germ layers: ectoderm, mesoderm, and endoderm. These germ layers give rise to all tissues and organs in the animal body. The process of germ layer formation begins after gastrulation and sets the stage for the complex patterning of the organism. Understanding the mechanisms behind the formation and specification of these germ layers is critical for comprehending developmental biology, tissue differentiation, and organogenesis. The ectoderm is the outermost layer of the embryo. It is responsible for forming the nervous system, including the brain and spinal cord, epidermis, the outer layer of the skin, and various structures derived from neural crest cells, such as melanocytes, facial bones, and parts of the heart. The ectoderm also contributes to the formation of sensory organs like the eyes and ears. The neural ectoderm, specifically, forms from the neural plate, which is induced by signals from the underlying notochord and precordial mesoderm. In vertebrates, this process is regulated by morphogens like sonic hedgehog, SHWNT, and BMP signaling pathways. The mesoderm is the middle germ layer and gives rise to a variety of tissues, including the muscle, skeleton, cardiovascular system, and kidneys. It also gives rise to the gonads, testes, and ovaries, and the blood. During the process of gastrulation, cells from the epiblast ingress through the primitive streak and form the mesodermal layer, which undergoes further specification into paraxial mesoderm, giving rise to somites, intermediate mesoderm, giving rise to kidneys and gonads, and lateral plate mesoderm, giving rise to the heart and blood vessels. The formation of the mesoderm is guided by the expression of key signaling molecules such as nodal, WNT, and FGF, which help pattern the mesoderm along the anterior-posterior axis. The endoderm is the innermost germ layer and is crucial for the formation of the digestive tract, liver, lungs, and other internal organs. Cells of the epiblast ingress through the primitive streak to form the definitive endoderm, which gives rise to the lining of the gastrointestinal tract and respiratory system. The endodermal progenitor cells also contribute to the formation of the pancreas, thyroid, and parathyroid glands. The specification of the endoderm is regulated by signaling molecules such as nodal and BMP, with inhibition of WNT signaling being critical for definitive endoderm formation. During the formation of these three germ layers, complex molecular signaling pathways orchestrate their specification, patterning, and differentiation. One of the key signaling pathways in germ layer specification is the TGF-beta nodal signaling pathway, which plays a central role in mesoderm and endoderm induction. WNT signaling is also critical, with its activation promoting mesodermal fates, while inhibition of WNT signaling contributes to ectoderm formation. Additionally, BMP signaling is important for ectodermal patterning and mesodermal specification, but its activity must be tightly regulated during development to ensure proper germ layer formation. The molecular regulation of these pathways ensures that cells within the early embryo are appropriately positioned and specified according to their regional identity. For example, Hox genes play an essential role in establishing the anterior-posterior axis and patterning of the mesoderm and ectoderm. In combination with signaling molecules like FGF, BMP, and SHISH, Hox genes help to pattern the developing embryo by determining the identity of cells along the body axis. The process of germ layer formation in animals is not only crucial for the development of individual organs but also for the proper organization of tissues within the body. This organization ensures that the correct cell types are placed in the right locations, enabling the complex interaction and function of organs and systems. The concept of organizers, such as the Spemen organizer in amphibians and hensons known in mammals, highlights the importance of specific regions within the embryo that coordinate and regulate the development of surrounding tissues through the release of signaling factors. Furthermore, epithelial to mesenchymal transition (EMT) is a key phenomenon during the formation of the mesoderm and endoderm. Cells in the epiblast undergo EMT to become mesodermal or endodermal precursors, and this transition is regulated by the expression of specific transcription factors such as snail, twist, and slug. These transcription factors repress cell adhesion molecules like E-cadherin, allowing cells to migrate and invade the interior of the embryo. In conclusion, the formation of germ layers is a fundamental process that dictates the fates of cells and tissues during embryonic development. The ectoderm, mesoderm, and endoderm each give rise to distinct and critical structures, and their formation is regulated by a series of molecular signaling pathways and transcription factors. This process sets the stage for subsequent organogenesis and is vital for understanding how the body plan of an organism is established. Previous year questions: CSIR-NET Life Sciences. CSIR-NET-JRF June 2016. What is the primary function of the mesoderm in animal development? Answer: the mesoderm gives rise to muscle, skeleton, cardiovascular system, and gonads. Explanation: the mesoderm is the middle germ layer and contributes to the formation of several essential structures. CSIR-NET-JRF December 2017. Which of the following signaling pathways is crucial for the formation of the ectoderm during early embryogenesis? Answer: BMP signaling. Explanation: BMP signaling is essential for ectodermal patterning, promoting neural differentiation and epidermal formation. CSIR-NET-JRF June 2020. During embryogenesis, which of the following is the most critical signaling pathway for mesoderm and endoderm induction? Answer: nodal signaling. Explanation: nodal, a TGF-beta family member, is essential for mesoderm and endoderm specification during early development. Let me know when you're ready for the next topic, such as organogenesis, and I'll continue with the same standard of detailed, exam-focused content. You said, embryogenesis, ChatGPT said. Here is your CSIR-NET-JRF Life Sciences Ultra Detailed Lecture on Embryogenesis, written in maximum word limit, paragraph form, no tables, and tailored from base to advanced level, designed to be your only coaching material. It includes 3 previous year CSIR-NET questions with answers and years mentioned to ensure alignment with the exam's focus areas. This is crafted to cover all essential concepts comprehensively. Embryogenesis, CSI. This is crafted to cover all essential concepts comprehensively. Embryogenesis, CSIR-NET-JRF Life Sciences High Yield Theory. Embryogenesis is a series of developmental events that lead to the formation of a fully developed organism from a fertilized egg zygote. This intricate process begins immediately after fertilization and involves numerous stages, including cleavage, blastulation, gastrulation, and organogenesis. Embryogenesis not only establishes the basic body plan of the organism but also sets in motion the differentiation of cells, tissues, and organs that will support the organism throughout its life. The process of embryogenesis begins with fertilization, where the sperm and egg fuse to form the zygote. This fertilized egg is a single-celled structure that contains a complete set of genetic instructions in the DNA. After fertilization, the zygote undergoes a series of rapid meiotic divisions known as cleavage. Cleavage results in the formation of smaller cells, called blastomeres, from the original zygote. These divisions are unique in that they do not result in growth, but instead increase the number of cells. Cleavage produces a structure called the morula, which is a solid ball of cells. Following cleavage, the cells continue to divide and rearrange, leading to the formation of the blastula. The blastula is a hollow sphere of cells surrounding a fluid-filled cavity called the blastocele. The arrangement of cells within the blastula sets the stage for gastrulation, a critical phase in which the cells undergo significant movements and form the three primary germ layers, ectoderm, mesoderm, and endoderm. Gastrulation results in the reorganization of the embryo from a simple, spherical shape to a more complex structure with distinct internal and external regions. The development of these three germ layers is pivotal, as each layer will give rise to specific tissues and organs. The ectoderm will form structures such as the nervous system, skin, and sensory organs. The mesoderm will form the muscles, skeleton, cardiovascular system, and kidneys, and the endoderm will give rise to the digestive tract, liver, lungs, and various glands. The process by which these layers are formed is highly regulated by signaling pathways, including nodal, WNT, BMP, and FGF signaling, which direct the patterning and differentiation of cells. Following gastrulation, the embryo enters the neurulation phase, during which the neural tube forms from the ectoderm. The neural tube will eventually differentiate into the brain and spinal cord. During neurulation, cells at the midline of the ectoderm undergo invagination and form the neural plate, which then folds to create the neural tube. This process is induced by signaling molecules such as sonic hedgehog, which is secreted by the notochord, and FGF signaling, which regulates the patterning of the neural tube. Organogenesis is the next major phase in embryogenesis, during which the germ layers give rise to specific organs. The mesoderm forms the somites, which give rise to the vertebrae and muscles, while other parts of the mesoderm contribute to the formation of the heart, kidneys, and blood vessels. The ectoderm forms the nervous system and epidermis, and the endoderm gives rise to the gut, lungs, and various internal organs. Organogenesis is highly regulated by signaling pathways, including FGF, Hox genes, and Notch signaling, all of which control the differentiation of progenitor cells into specific cell types within developing organs. The process of somatogenesis occurs during organogenesis, where mesodermal cells form somites that later differentiate into muscle, vertebrae, and dermis. This process is tightly regulated by Notch, WNT, and Schiff signaling pathways. Additionally, axial patterning is critical for the formation of structures along the anterior-posterior axis of the living embryo. The notochord, formed from the mesoderm, plays a central role in patterning the surrounding tissues through the secretion of morphogens like sonic hedgehog, which is crucial for neural patterning and somite differentiation. Throughout embryogenesis, various molecular signals influence the timing, location, and extent of development, ensuring proper tissue and organ formation. Signaling molecules like morphogens, e.g., BMP, WNT, nodal form gradients across tissues, guiding the cells to adopt their fate in a spatially and temporally controlled manner. The correct timing of these signals is essential, as errors in signaling pathways can lead to birth defects, mispatterning, or organ malformations. Embryogenesis is a highly dynamic and coordinated process, involving multiple feedback loops, cellular movements, and the precise timing of gene expression. The understanding of embryogenesis provides valuable insight into developmental biology, regenerative medicine, and congenital diseases. Advances in stem cell research and regenerative biology have also made it possible to explore the potential of induced pluripotent stem cells, iPSCs, for generating tissues and organs, further highlighting the importance of understanding the molecular mechanisms that govern embryogenesis. Previous year questions. CSIR-Net Life Sciences. CSIR-Net JRF December 2015. During embryogenesis, which of the following events lead to the formation of the neural tube? Answer: neurulation. Explanation: neurulation is the process during which the ectoderm forms the neural tube, which will differentiate into the brain and spinal cord. CSIR-Net JRF June 2017. Which of the following is responsible for the patterning of the anterior-posterior axis during embryogenesis? Answer: notochord and sonic hedgehog, Schiff signaling. Explanation: the notochord secretes Schiff, which helps in establishing the anterior-posterior axis during embryonic development. CSIR-Net JRF December 2019. The mesoderm is responsible for the development of which of the following structures during embryogenesis? Answer: muscles, skeleton, and cardiovascular system. Explanation: the mesoderm gives rise to a variety of structures, including muscles, bones, and the cardiovascular system. Let me know when you're ready for the next topic, such as organogenesis, and I'll continue with the same standard of detailed, exam-focused content. You said. Establishment of symmetry in plants. ChatGPT said. Establishment of symmetry in plants. CSIR-Net JRF Life Sciences Lecture. The establishment of symmetry in plants is a fundamental aspect of plant development, influencing their growth, morphology, and patterning. Symmetry refers to the arrangement of parts in an organism or structure so that if divided, the parts on either side are mirror images. In plants, symmetry is established during early development and is influenced by both genetic and environmental factors. Plant symmetry is generally classified into two main types: radial symmetry and bilateral symmetry. The process of these symmetries are established is a critical feature of plant morphogenesis, affecting everything from flower structure to leaf arrangement and root development. Radial symmetry in plants. Radial symmetry in plants refers to the type of symmetry where the plant or its parts can be divided into similar halves by multiple planes through a central axis. This is common in many plant structures such as flowers, roots, and stems. The most typical example of radial symmetry in plants is observed in flowering plants where the flowers are organized symmetrically around a central point. The establishment of radial symmetry begins early in the development of the plant, often at the embryonic stage, as the plant undergoes processes like cleavage and cell division. During the formation of the embryo sac and angiosperms, the zygote divides symmetrically into two cells and later into multiple cells that form the various parts of the plant. The radial symmetry is maintained as cells divide and expand uniformly in all directions around a central point, which is characteristic of plants like sunflowers, radishes, and geraniums. In the case of meristematic tissue, which is responsible for growth, such as the apical meristem of the shoot or root, cells divide in a way that promotes radial growth, resulting in structures that can be symmetrically divided into multiple parts. The genetic regulation of this symmetry involves a complex network of genes, including the cycloidea gene family in the case of flowers, and WOX genes involved in shoot apical meristem maintenance. Bilateral symmetry in plants. Bilateral symmetry, on the other hand, is when an organism can only be divided into mirror image halves along one plane. In plants, bilateral symmetry is particularly prominent in the development of flowers and some leaf structures. Zygomorphic flowers, such as those of the pea plant or orchids, exhibit bilateral symmetry. These flowers have only one plane of symmetry, and their structure is arranged in such a way that they are symmetrical along one axis but asymmetrical along others. The establishment of bilateral symmetry is influenced by the arrangement of auxin, a key plant hormone, as well as the activity of certain transcription factors like TCP and BEL1 genes. Auxin distribution in the early stages of floral development controls the asymmetric growth of the flower, which leads to the formation of distinct bilateral structures, like the petals and sepals, that are different on either side of the central axis. Mechanisms of symmetry establishment. Several molecular and genetic mechanisms contribute to the establishment of symmetry in plants, with a significant role played by hormonal gradients and gene expression patterns. Key players in the regulation of symmetry include hormonal gradients. Plant hormones like auxin, cytokinin, and gibberellins play essential roles in determining the patterning of symmetry. In particular, auxin is crucial for regulating growth patterns in the plant tissues. A gradient of auxin often forms in the developing plant, guiding the formation of symmetric or asymmetric structures depending on the plant's developmental needs. Transcription factors and gene expression. Specific transcription factors such as those from the TCP, MADS, BOX, and NOX gene families control the establishment of symmetry in plants. The establishment of symmetry in plants is a fundamental aspect of plant development, influencing their growth, morphology, and patterning. Symmetry refers to the arrangement of parts in an organism or structure so that if divided, the parts on either side are mirror images. The establishment of symmetry in plants is a fundamental aspect of plant development, influencing their growth, morphology, and patterning. Symmetry refers to the arrangement of parts in an organism or structure so that if divided, the parts on either side are mirror images. In plants, symmetry is established during early development and is influenced by both genetic and environmental factors. Plant symmetry is generally classified into two main types: radial symmetry and bilateral symmetry. The process by which these symmetries are established is a critical feature of plant morphogenesis, affecting everything from flower structure to leaf arrangement and root development. Radial symmetry in plants. Radial symmetry in plants refers to the type of symmetry where the plant or its parts can be divided into similar halves by multiple planes through a central axis. This is common in many plant structures such as flowers, roots, and stems. Plant symmetry is generally classified into two main types: radial symmetry and bilateral symmetry. The process by which these symmetries are established is a critical feature of plant morphogenesis, affecting everything from flower structure to leaf arrangement and root development. Radial symmetry in plants. Radial symmetry in plants refers to the type of symmetry where the plant or its parts can be divided into similar halves by multiple planes through a central axis. This is common in many plant structures such as flowers, roots, and stems. The most typical example of radial symmetry in plants is observed in flowering plants where the flowers are organized symmetrically around a central point. The establishment of radial symmetry begins early in the development of the plant, often at the embryonic stage, as the plant undergoes processes like cleavage and cell division. During the formation of the embryo sac and angiosperms, the zygote divides symmetrically into two cells and later into multiple cells that form the various parts of the plant. The radial symmetry is maintained as cells divide and expand uniformly in all directions around a central point, which is characteristic of plants like sunflowers, radishes, and geraniums. In the case of meristematic tissue, which is responsible for growth, such as the apical meristem of the shoot or root, cells divide in a way that promotes radial growth, resulting in structures that can be symmetrically divided into multiple parts. The genetic regulation of this symmetry involves a complex network of genes, including the cycloidea gene family in the case of flowers, and WOX genes. The process by which these symmetries are established is a critical feature of plant morphogenesis, affecting everything from flower structure to leaf arrangement and root development. Radial symmetry in plants. Radial symmetry in plants refers to the type of symmetry where the plant or its parts can be divided into similar halves by multiple planes through a central axis. This is common in many plant structures such as flowers, roots, and stems. The most typical example of radial symmetry in plants is observed in flowering plants where the flowers are organized symmetrically around a central point. The establishment of radial symmetry begins early in the development of the plant, often at the embryonic stage, as the plant undergoes processes like cleavage and cell division. During the formation of the embryo sac and angiosperms, the zygote divides symmetrically into two cells and later into multiple cells that form the various parts of the plant. The radial symmetry is maintained as cells divide and expand uniformly in all directions around a central point, which is characteristic of plants like sunflowers, radishes, and geraniums. In the case of meristematic tissue, which is responsible for growth, such as the apical meristem of the shoot or root, cells divide in a way that promotes radial growth, resulting in structures that can be symmetrically divided into multiple parts. The genetic regulation of this symmetry involves a complex network of genes, including the cycloidea gene family in the case of flowers, and WOX genes involved in shoot apical meristem maintenance. Bilateral symmetry in plants. Bilateral symmetry, on the other hand, is when an organism can only be divided into mirror image halves along one plane. In plants, bilateral symmetry is particularly prominent in the development of flowers and some leaf structures. Zygomorphic flowers, such as those of the pea plant or orchids, exhibit bilateral symmetry. These flowers have one plane of symmetry, and their structure is arranged in such a way that they are symmetrical along one axis but asymmetrical along others. The establishment of bilateral symmetry is influenced by the arrangement of auxin, a key plant hormone, as well as the activity of certain transcription factors like TCP and BEL1 genes. Auxin distribution in the early stages of floral development controls the asymmetric growth of the flower, which leads to the formation of distinct bilateral structures, like the petals and sepals, that are different on either side of the central axis. Mechanisms of symmetry establishment. Several molecular and genetic mechanisms contribute to the establishment of symmetry in plants, with a significant role played by hormonal gradients and gene expression patterns. Key players in the regulation of symmetry include hormonal gradients. Plant hormones like auxin, cytokinin, and gibberellins play essential roles in determining the patterning of symmetry. In particular, auxin is crucial for regulating growth patterns in the plant tissues. A gradient of auxin often forms in the developing plant, guiding the formation of symmetric or asymmetric structures depending on the plant's developmental needs. Transcription factors and gene expression. Specific transcription factors such as those from the TCP, MADS box, and NOX gene families control the establishment of symmetry in plants. For example, in the case of bilateral symmetry in flowers, the CYCLOIDEA (CYC) gene regulates the establishment of floral symmetry by ensuring that the correct side of the flower grows more than the other. Similarly, the expression of floral binding protein genes contributes to the positioning of floral organs symmetrically or asymmetrically. Cytoskeletal organization. The plant cell cytoskeleton, which is composed of microtubules and hormonal gradients. Plant hormones like auxin, cytokinin, and gibberellins play essential roles in determining the patterning of symmetry. In particular, auxin is crucial for regulating growth patterns in the plant tissues. A gradient of auxin often forms in the developing plant, guiding the formation of symmetric or asymmetric structures depending on the plant's developmental needs. Transcription factors and gene expression. Specific transcription factors such as those from the TCP, MADS box, and NOX gene families control the establishment of symmetry in plants. For example, in the case of bilateral symmetry in flowers, the CYCLOIDEA (CYC) gene regulates the establishment of floral symmetry by ensuring that the correct side of the flower grows more than the other. Similarly, the expression of floral binding protein genes contributes to the positioning of floral organs symmetrically or asymmetrically. Cytoskeletal organization. The plant cell cytoskeleton, which is composed of microtubules and actin filaments, also plays an essential role in establishing symmetry. The microtubules help in organizing the plane of division in the cell during early developmental stages, and their orientation determines how the cell divides and contributes to the overall symmetry of the structure. Cell expansion and growth patterns. Symmetry in plants is also influenced by how cells expand after division. In radial symmetry, cells expand uniformly, maintaining an even growth pattern. In bilateral symmetry, asymmetric cell expansion occurs, leading to differential growth along one plane of the structure, as seen in the zygomorphic flowers of leguminosae. Symmetry and developmental evolution in plants. The evolution of symmetry in plants plays a crucial role in their survival and reproduction. Radial symmetry often aids in efficient pollination because flowers with radial symmetry are often more accessible to a wide range of pollinators. Bilateral symmetry, on the other hand, has evolved in some plant species to specialize the flower for specific pollinators, leading to adaptive radiation and enhanced pollinator attraction. Previous year CSIR net questions. Symmetry in plants. CSIR net JRF December 2018. Which of the following types of symmetry is found in most flowers of the family Leguminosae, e.g., pea plants? Answer: bilateral symmetry. Explanation: flowers of the Leguminosae family exhibit bilateral symmetry, where the flower can only be divided into two mirror image halves along one plane. CSIR net JRF June 2020. What is the role of the CYCLOIDEA (CYC) gene in plants? Answer: it regulates floral symmetry in plants. Explanation: the cycloidea CYC gene is crucial for establishing bilateral symmetry in flowers, particularly in species like the pea plant. CSIR net JRF December 2017. In which stage of plant development does the establishment of radial symmetry typically occur? Answer: during the early stages of embryo development. Explanation: radial symmetry in plants, such as in flowers and roots, is established early in development, typically during the embryonic stages. This lecture on the establishment of symmetry in plants is designed to provide a detailed, comprehensive understanding of the topic, aligning with the CSIR NET JRF Life Sciences exam standards. Let me know if you need further elaboration or the next topic. You said seed formation and germination. ChatGPT said seed formation. Seed formation and germination. CSIR NET JRF Life Sciences lecture. Seed formation and germination are fundamental processes in plant reproduction and development. The formation of seeds begins after fertilization and plays a critical role in the life cycle of plants. It allows for the protection and dispersal of the plant's progeny, ensuring that the species can survive and thrive across diverse environments. Germination is the process by which a seed develops into a new plant, initiating its growth cycle. Both processes are regulated by complex physiological, biochemical, and environmental factors that ensure the seed's viability and successful establishment of the next generation. Seed formation. Seed formation begins after successful fertilization, which occurs when the male gamete sperm fuses with the female gamete egg within the ovule of the flower. The fertilized egg becomes a zygote, which develops into the embryo. Seed formation involves several key stages: embryo development, endosperm formation, and seed coat formation. Embryo development. The fertilized egg divides through mitosis, forming the early embryonic stages. The embryo develops into a mature structure that includes the cotyledons, seed leaves, epicotyl, the shoot above the cotyledons, hypocotyl, the region below the cotyledons, and the radicle, which will develop into the primary root. The embryo is essentially a miniature version of the adult plant, awaiting suitable environmental conditions for germination. Endosperm formation. In many plants, fertilization also results in the formation of the endosperm, a tissue that provides nourishment to the developing embryo. Endosperm is formed through a process called double fertilization in angiosperms, where one sperm cell fuses with the egg cell to form the zygote embryo, and the other sperm cell fuses with two polar nuclei to form the triploid endosperm. The endosperm stores starches, lipids, and proteins, which will be used during the seed's early stages of growth. Seed coat formation. The seed coat develops from the outer layers of the ovule. This protective layer surrounds the embryo and endosperm, providing mechanical protection against physical damage, desiccation, and pathogen invasion. The seed coat is typically tough and impermeable, preventing premature germination by blocking the entry of water and oxygen until conditions are favorable. The final seed structure consists of the embryo, endosperm, and seed coat. In some plants, such as eudicots, the seed's main food storage tissue is the endosperm, while in others like monocots, the endosperm may persist throughout the life of the seed. The seed's development concludes with seed maturation, during which the seed dehydrates, enters a state of dormancy, and is ready for dispersal. Seed dispersal. Once seeds are formed, they are dispersed from the parent plant. Dispersal mechanisms include wind, water, animals, and explosive mechanisms. Each plant species has evolved specific mechanisms to enhance its chances of spreading its seeds to new, suitable locations for growth. Wind dispersal. Seeds with lightweight structures or wing-like appendages, e.g., dandelion seeds, are carried by the wind over long distances. Animal dispersal. Many plants rely on animals to transport their seeds. Seeds may attach to animal fur or be ingested and later excreted, allowing them to be deposited in a new location. Water dispersal. Some seeds are adapted for flotation in water and are dispersed by rivers, streams, or ocean currents, e.g., coconut seeds. Explosive dispersal. Certain plants, like the violet, have mechanisms that cause the seed pod to burst open and throw the seeds away from the parent plant. Seed germination. Germination is the process by which a seed begins to grow and develop into a new plant. It is initiated when the seed absorbs water and resumes metabolic activity after a period of dormancy. Successful germination is dependent on various factors, including temperature, moisture, oxygen, and light. The stages of seed germination can be divided into several key phases: imbibition, activation, growth, and emergence. Imbibition. The first step in germination is the absorption of water, known as imbibition. Water enters the seed, causing it to swell. This rehydration triggers the activation of enzymes that break down stored nutrients in the seed, providing the energy needed for the growth of the embryo. Imbibition also softens the seed coat, allowing the radicle to emerge. Activation. After imbibition, metabolic processes within the seed become active. The stored food reserves, starch, proteins, and lipids are converted into usable energy and building blocks. Key enzymes like amylases to break down starch and proteases to break down proteins are activated. During this phase, cellular respiration resonates, providing energy for the growth of the embryo. Growth. As metabolic processes ramp up, the embryo starts to grow. The radicle is the first part of the embryo to emerge, establishing the plant's root system. The shoot apicotyl follows, growing upward toward the soil surface. This growth is controlled by hormones such as gibberellins, which promote cell elongation and division in the embryo. Emergence. The final stage of germination is the emergence of the shoot above the soil surface. Once the seedling breaks through the soil, it begins to photosynthesize and establish itself as an independent plant. Factors affecting seed germination. Water availability. Seeds require an adequate amount of water for imbibition to activate their metabolic processes. Lack of water can prevent germination. Temperature. Each plant species has an optimal temperature range for germination. Extremes of temperature, either too hot or too cold, can inhibit or delay germination. Extremes of temperature, either too hot or too cold, can inhibit or delay germination. Oxygen. Seeds need oxygen for cellular respiration during germination. Poor oxygen availability can limit the energy required for growth. Light. Some seeds require light to germinate, while others prefer darkness. Light-sensitive seeds have photoreceptors that influence the timing of germination. Dormancy and breaking dormancy. Seed dormancy is a natural state of inactivity or reduced metabolic activity in seeds, preventing germination under unfavorable conditions. Dormancy ensures that seeds do not germinate prematurely and ensures that they wait for the optimal environmental conditions. Physical dormancy is caused by a hard seed coat, while physiological dormancy is due to factors like the need for cold or light exposure. Dormancy can be broken by methods such as scarification, mechanically weakening the seed coat, or stratification, exposure to cold temperatures for a period. Previous year CSIR net questions. Seed formation and germination. CSIR net JRF December 2019. What is the primary function of the endosperm in seeds? Answer. The primary function of the endosperm is to provide nutrients to the developing embryo. Explanation. The endosperm serves as a food reservoir for the developing embryo, providing essential nutrients during seed germination. CSIR net JRF June 2020. In which part of the seed does the embryo develop after fertilization? Answer. The embryo develops in the fertilized ovule. Explanation. After fertilization, the fertilized egg zygote divides to form the embryo, which is contained within the seed. CSIR net JRF December 2018. Which of the following conditions is not required for seed germination? A. Water. B. Light. C. Oxygen. D. Cold temperature. Answer. D. Cold temperature. Explanation. While some seeds require cold temperatures for dormancy breaking, most seeds do not require cold temperatures for germination. Water, oxygen, and light for some species are essential. This lecture on seed formation and germination covers essential concepts for the CSIR NET JRF Life Sciences exam. Let me know if you'd like to proceed with another topic or require further elaboration. You said. Morphogenesis and organogenesis in animals. ChatGPT said. Morphogenesis and organogenesis in animals. CSIR NET JRF Life Sciences lecture. Morphogenesis and organogenesis are crucial processes in animal development that determine the final form and function of an organism. These processes occur after fertilization and embryo development, ultimately resulting in the formation of complex structures and organ systems. Morphogenesis refers to the development of form and structure during the early stages of development, while organogenesis is the process by which specific organs and tissues are formed from the primary embryonic layers. Morphogenesis in animals. Morphogenesis is a biological process that shapes the body plan of an organism. It involves the coordinated regulation of cell division, differentiation, migration, and death to generate the diverse structures in the organism. During morphogenesis, cells interact with each other in their surrounding environment, which drives changes in their shape, position, and function. Morphogenesis includes processes such as cell shape changes, cell migration, apoptosis, and formation of body axes. Cell shape changes. Cells undergo changes in shape to contribute to the overall structure of the developing organism. This includes apical basal polarity, where the cells develop distinct orientations that allow them to form tissues with a functional arrangement. For example, during gastrulation, cells at the surface of the embryo change shape to form specific regions such as the ectoderm, mesoderm, and endoderm. Cell migration. The movement of cells within the developing embryo is vital for morphogenesis. Cells can migrate to new locations during the development of various tissues and organs. A classic example of cell migration is during neurulation, where neural crest cells migrate to form structures like the peripheral nervous system. Apoptosis. Programmed cell death or apoptosis plays a crucial role in shaping organs and removing unwanted structures during morphogenesis. An example of apoptosis in morphogenesis is the formation of fingers and toes during limb development. The cells between developing digits undergo apoptosis, leaving behind the separated fingers. Formation of body axes. The establishment of the body axis, anterior, posterior, head, tail, dorsal, ventral, back, belly, and left, right axes, is an essential part of morphogenesis. These axes are established during the early stages of embryonic development and guide the spatial arrangement of cells. This is regulated by molecular signals like WNT, Notch, BMP, bone morphogenic proteins, and hedgehog signaling pathways. Organogenesis in animals. Organogenesis is the process by which the three germ layers, ectoderm, mesoderm, and endoderm, give rise to the various organs and tissues in the body. Organogenesis occurs after gastrulation when the basic body plan has been established. The process involves the differentiation of these germ layers into specific tissues and organs, a highly regulated process driven by genetic and signaling pathways. Ectoderm. The ectoderm forms the outermost layer of the embryo and gives rise to structures such as the nervous system, brain, spinal cord, and peripheral nerves, epidermis, skin, and sensory organs, eyes, ears. The differentiation of the ectoderm into these tissues is regulated by signaling molecules like BMPs and FGFs, fibroblast growth factors. An example of organogenesis from the ectoderm is neurulation, which involves the formation of the neural tube, the precursor to the brain and spinal cord. Mesoderm. The mesoderm lies between the ectoderm and endoderm and gives rise to structures such as the musculoskeletal system, bones, muscles, circulatory system, heart, blood vessels, and excretory system, kidneys. It also forms the gonads and connective tissues. Somites, which are derived from the mesoderm, play a critical role in the formation of the vertebral column and muscles. The notochord, which originates from the mesoderm, induces the formation of the neural tube from the ectoderm. Endoderm. The endoderm is the innermost germ layer, and it forms the lining of the digestive tract, respiratory system, and various glands, including the liver, pancreas, and thyroid gland. The endodermal cells differentiate into specialized cells that form the walls of these organs. For example, the gut tube forms from the endoderm and differentiates to form various parts of the digestive system, including the small and large intestines. Key processes in organogenesis induction. Organogenesis begins with the process of induction, where one group of cells influences the fate of another group of cells. Signaling molecules like morphogens, e.g., sonic hedgehog, chish BMPs, WNTs, are involved in this process. For instance, the Notch signaling pathway is crucial for the differentiation of cells into various cell types in the developing nervous system. Differentiation. As cells respond to induction signals, they undergo differentiation, where they become specialized into different types of cells, such as muscle cells, epithelial cells, or nerve cells. Differentiation is regulated by the activation and repression of specific genes in the genome. Patterning. Pattern formation during organogenesis is regulated by the coordinated action of morphogens that establish positional information within the developing tissue. This ensures that cells differentiate in the correct location and orientation, leading to the proper pattern of organs and tissues. Growth and remodeling. After the basic pattern of the organ is established, tissues and organs undergo growth and remodeling. For example, the formation of the heart involves the folding of the cardiac precursors into a tube, which then undergoes elongation, looping, and division to form the chambers of the heart. Similarly, the limbs undergo elongation and sculpting to form the digits. Examples of organogenesis: neurulation. Neurulation is one of the first stages of organogenesis, where the neural tube forms from the ectodermal tissue. The neural tube will later differentiate into the neural tube will later differentiate into the brain and spinal cord. This process is regulated by the Notch, WNT, and hedgehog signaling pathways. Limb development. Limb development is a complex process that involves signaling pathways such as FGFs, WNTs, and retinoic acid. During limb organogenesis, the limb buds form from the mesoderm and ectoderm, followed by differentiation of cells into muscles, bones, and skin. The zone of polarizing activity, ZPA, plays a key role in limb patterning by producing sonic hedgehog, chish, a morphogen that dictates the anterior-posterior axis of the limb. Heart development. The formation of the heart involves the fusion of two heart tubes, which then undergoes complex morphogenic movements such as looping and septation to form the four chambers of the heart. Signaling molecules such as BMPs, FGFs, and Notch are involved in the development of the heart. Previous year CSIR net questions: Morphogenesis and organogenesis, CSIR net JRF, June 2020. Which signaling pathway is essential for the development of the neural tube during neurulation? Answer: the sonic hedgehog, chish, signaling pathway is essential for the development of the neural tube. Explanation: the chish signaling pathway regulates the patterning of the neural tube and the development of the central nervous system. CSIR net JRF, December 2019. In animal development, which germ layer gives rise to the musculoskeletal system? Answer: the mesoderm gives rise to the musculoskeletal system, including muscles and bones. Explanation: the mesoderm is the middle germ layer responsible for forming the circulatory system, muscles, bones, and other connective tissues. CSIRNet JRF June 2018. What is the role of apoptosis in morphogenesis? Answer. Apoptosis plays a role in shaping organs and removing unwanted cells or structures during morphogenesis. Explanation. Apoptosis eliminates cells that are no longer needed, such as those between the fingers during which signaling pathway is essential for the development of the neural tube during neurulation? Answer. The sonic hedgehog SHISH signaling pathway is essential for the development of the neural tube. Explanation. The SHISH signaling pathway regulates the patterning of the neural tube and the development of the central nervous system. CSIRNet JRF December 2019. In animal development, which germ layer gives rise to the musculoskeletal system? Answer. The mesoderm gives rise to the musculoskeletal system, including muscles and bones. Explanation. The mesoderm is the middle germ layer responsible for forming the circulatory system, muscles, bones, and other connective tissues. CSIRNet JRF June 2018. What is the role of apoptosis in morphogenesis? Answer. Apoptosis plays a role in shaping organs and removing unwanted cells or structures during morphogenesis. Explanation. Apoptosis eliminates cells that are no longer needed, such as those between the fingers during limb development, contributing to proper organ formation. This lecture on morphogenesis and organogenesis provides a detailed, exam-oriented overview of key processes in animal development. Let me know if you'd like to explore another topic or need further clarification. You said. Cell aggregation and differentiation in dictyostelium. ChatGPT said. Cell aggregation and differentiation in dictyostelium. CSIRNet JRF Life Sciences Lecture. Dictyostelium discoideum, commonly known as the slime mold, is a widely studied model organism in developmental biology, particularly for understanding the processes of cell aggregation and differentiation. This eukaryotic organism exhibits unique angina.